In a study using specialized imaging techniques, Johns Hopkins Medicine researchers report distinctive changes in the white matter and other brain tissue physiology of those with post-treatment Lyme disease, a condition affecting 10% to 20% of the nearly 500,000 Americans who contract Lyme. The study's findings, published October 26 in the journal PLOS ONE, substantiate and help validate that memory and other cognitive difficulties experienced long-term by individuals with post-treatment Lyme disease are linked to functional and structural changes in the brain. Lyme disease, whose early symptoms may include a characteristic rash, flu-like aches and fever, joint pain, and fatigue, is treated using a rigorous course of antibiotics, which usually clears the illness. Such individuals generally have no obvious clinical or laboratory evidence of ongoing issues. In a bid to identify what they suspect are long-term changes in brain function that may be causing certain persistent Lyme disease symptoms, researchers from Johns Hopkins Medicine's Departments of Neurology and Psychiatry and Behavioral Sciences, in collaboration with the Lyme Disease Research Center. These scans allow investigators to track changes in the brain in real time. Objective biologic measures of post-treatment Lyme symptoms typically can't be identified using regular MRIs, CT scans, or blood tests, says researcher John Aucott, MD, director of the Johns Hopkins Lyme Disease Clinical Research Center and associate professor of medicine at the Johns Hopkins. We needed to expand our methods of evaluation, he adds. Participants were asked to memorize and recall capital and lowercase letters, as well as the alphabetical order of multiple letters. White matter is crucial for moving information around the brain, acting like train tracks that help deliver information to the train depot, or gray matter. We saw certain areas in the frontal lobe underactivating and others that were overactivating, which was somewhat expected, says Marvel, an associate professor of neurology. This unique approach corroborated their fMRI findings and revealed new findings. Water was diffusing, or leaking, along the patient's axons, the extensions of neurons that carry electrical signals to other neurons, within the same white matter regions identified in the fMRI. The researchers believe the increased activity they saw in white matter may reflect a helpful immune response in the post-treatment Lyme patients, as it correlated with better disease outcomes. However, this finding was also correlated to the patients with post-treatment Lyme needing longer periods of time to complete the memory task. The researchers say this is indicative that the white matter response could be an adaptive one, but that the white matter's physiology and function have been changed, which could come at a cost. Approaching this problem collaboratively with experts in different fields is what gave us these novel findings, and will hopefully answer our remaining questions, specifically about the role of axonal leakage and white matter activation in post-treatment Lyme disease, she says. Marvel and Aucott say this research may help illuminate underlying mechanisms and potential therapeutic targets for neurologic Lyme disease. Their findings may also have relevance to other kinds of infection-associated chronic illnesses, including long COVID and chronic fatigue syndrome. Funding for the project was provided by an anonymous donor to John Aucott. The research team also includes Kylie Dia Bhattacharya, Alison Rebman, Arnold Bacher, Jason Creighton, Erica Koziro, Arun Venkatasan, and Priyanka Nadkarni of Johns Hopkins Medicine, as well as Owen Morgan, who is now at Cornell University. https://ift.tt.nmvyadr. Accessed October 26, 2022. Thanks for watching. Please subscribe my channel for more videos.